Hello, my name's Gabby, and today I will be talking about unveiling bat immunology through toll-like receptor analysis. So, bats possess this interesting ability to fight off viruses, and really they suppress viruses. They still have it in their body, but they don't show symptoms like humans do. And one of the factors that play into this is toll-like receptors, which are these proteins that are kind of shaped like horseshoes or maybe even half of a horseshoe. And they play a role in the immune system's first line of defense. And they fight off things such as bacteria, fungi, and viruses. And they're found on the cell membrane as well as the endosomes. So the goal of this study was to delve more deeper into studying toll-like receptors so that in bats, so that we can then replicate it and have an immune system that responds to viruses as strongly as bats do for humans. So this study specifically focuses on viral DNA as well as viral RNA. And the specific toll-like receptors in these various bat species that were looked at was TLR1, 2, 3, 6, 7, 8, and 9, as these were found to respond to viruses. And we can see here in this diagram right here that one distinction between bats and humans is that normally when a human gets a virus, it causes inflammation, whereas in bats, they dampen inflammation, which can then also be a key factor into why bats are able to suppress this viral activity more compared to humans. And to just give more of a brief overview of TLRs, we have right here TLR4, and this is a respiratory synthetic virus, and this virus is docking onto TLR4, and this leads to TLR4 detecting the viral protein. Then this virus infects the host cell, and then this viral DNA. This viral DNA then gets recognized by TLR9. Then after that, it goes through a signaling process, a pathway to be more specific with pathway soon leads to the cell recognizing the virus and triggering a immune response, and this immune response gets cells sent to adjacent cells, which is parasignaling. So my methods involved using gene sequences from the NCBI gene database, and I put this through MAFT, which is an alignment tool that will align the sequences that I gave it, and then I put that through IQ-tree to get phylogenetic trees, and then as also ITOL to label said trees. Then I used the protein sequences of the TLRs 1, 2, 3, 6, and so on and so forth to then make an AlphaFold 3 models. And on the topic of phylogenetic trees, right here, this shows various species of TLR6, and this just highlights the commonalities that we see with bat species with the same genus being having similar TLR sequences, so we could say that they have similar TLR structures. And this kind of highlights the evolutionary history shared between all mammals and TLR adaptations. And then right here with this graph, this shows the number of exons with TLRs 3, 7, and 8. And exons are actually coding regions of the protein. So these are the regions that code for the TLR3, TLR7, and TLR8. And as we can see with the common theme, for the most part, bat species have a higher number of exons than humans, which are in the green. So this could help explain maybe that one reason why bat TLRs are so advanced or are able to suppress viral activity is because they have a higher exon count. Then finally, with AlphaFold, this figure 4 is TLR8 in humans, and this is as well as COVID-19. There's kind of limited interaction with this TLR. But when we look here with TLR2, and this is a bat virus, and this is TLR2 in bats, or this is TLR2 in vampire bats. And as we can see here, that the virus kind of goes through the TLR channel and it binds a little bit better. So the TLR2 in this bat is a little more effective at recognizing the virus, just seen by its binding in the alpha fold compared to TLR8 in humans with the COVID virus. In conclusion, I compared the TLR nu uh, nucleic acid sequences in various bat species, made a phylogenetic tree, and in future research, I hope that it's possible to delve more deeper into the functional mechanisms of TLRs in bats, like the molecular processes that goes through, and aims to identify specific adaptations that we can mimic in human cells.